this we will discuss about this FCPR03 that helps in blocking the neural damage. And what people have think is that it could work in the similar way the PDE4 inhibition works. So what PDE4 inhibition that we discussed in the last couple of video, that it will reduce the ER stress through the through the increase in the amount of NRF inside the nucleus, that will produce oxyhomogenase 1 that will prevent the ER stress by neutralizing the ROS or reactive oxygen species. So people might have think that since the final objective of this FCRP03 matches with the PDE4 inhibition, and FCPR03 could have worked the similar way that the PDE4 works. So what they have done is they have first checked whether the proteins that are involved in ER stress get changed whenever the cells are treated with FCPR03 or not. So they have used the Western blot to check this. So they have used different conditions. First one is the negative control, where there is no ER stress, artificially ER stress generated, and there is no FCPR03. And then they have introduced FCPR03 and there is not much change. And then another condition in which they have used the ER stress generated as is oxygen glucose deprivation, and there also it seems that there is slight increase in the GRP 78. Then they have used both the condition, that is treat the cells with FCRP03 for 1 hour and then put the cells in oxygen glucose deprivation for 6 hours. Then they have seen that there is a slight decrease in the GRP 78 compared to the A. And similar thing they did for other proteins, that is phosphorylated PERK. We can see that in case of only ER stress, the phosphorylated PERK is increased to a greater extent, and when cells are pre-treated with FCPR03, there is a slight decrease in the phosphorylated PERK. And same thing has been observed with the phosphorylated EIF2 alpha. It is less when the cells are pre-treated with FCPR03. So then they did is they quantified these proteins using semi-quantitative analysis of Western blot. They found that there is a decrease in the GRP 78, there is a decrease in the phosphorylated PERK, is phosphorylated EF2 alpha. The CHOP proteins also that have missed is also reduced whenever the cells are pre-treated with FCPR03, and then ER stress is introduced, and same is shown in the semi-quantitative analysis of the Western blot. So all this shows that all this protein analysis shows that there is a decrease in the ER stress whenever the cells are pre-treated with FCPR03. So it might work similar way the PDE4 inhibition works. And they have also made this fluorescence image, in which we can see that when the cells are treated with oxygen glucose deprivation and without any pretreatment of FCPR03, there is a increase in the amount of this phosphorylated PEF2 alpha, and when the cells are pretreated with FCP0R3, there is a decrease in the amount of phosphorylated E alpha. And this is the control, DAPI is the control. We can see that the amount of DAPI remains same, and this is the ratio of both of them. We can see that the ratio in which cells are pre-treated with FCPR03 has less phosphorylated EIF2 alpha. So this could be one of the indication that there is less ER stress. So next they did is they check another condition that occurs in the cells when there is an inhibition of PDE4 in the cells. That is, there is a translocation of this NRF2 into the nucleus increases. So that they did the experiment to check whether there is an increase in the amount of NRF2 within the nucleus or not. So in order to check this, they stained the NRF2 and measured the intracellular location with the help of confocal microscopy. You can see that when the cells are treated with FCPR03, there is an increase in the amount of NRF2, and similarly they have seen that there is an increase in the amount of NRF2 when the cells are treated with FCRP03 and the ER stress is there, though it is lower than when there is no ER stress. This is one good reason could be that ER stress might affect the stabilization of this NRF2 or by directly or indirectly. And when the cells are treated with only with the ER stress without any FCPR03, there is less number of NRF2. Then they need to check whether this NRF2 is present in the cytoplasm, or it is present in the whether the NRF2 levels are more in the cytoplasm or in the nucleus. They did the experiment first at the cytoplasm and second at the nucleus, and so we can see that in the cytoplasm there is less number of NRF2 when there is no pretreatment of FCPR03. However, when the cells are pretreated with FCPR03, there is an increase in the amount of NRF2. And same things has been observed in the nucleus. There is an increase in the amount of NRF2 when the cells are pretreated with FCPR03. So, and then they check. Then they did the semi-quantitative analysis of this Western blot, and they find out that there is an increase in the amount of NRF2 in the cytoplasm, and also there is an increase in the amount of NRF2 in the nucleus. And when they did the nuclear staining of this, when they did the quantitative analysis of this confocal microscopy through image J, and then they also find out that the amount of NRF2 levels in the cells that are treated with FCPR03 before experiencing the ER stress to oxygen glucose deprivation has more NRF2 levels. So this could indicate that FCPR03 will promote, or we can say upregulate this NRF2 production and into the nucleus, that will lead to more OH1 generation, or we can say HO generation, that is heme-oxygenase 1 generation, that will neutralize ROS, thus it will trigger less ER stress as less amount of ROS is present in the cells. So this is how the FCPR03 will be able to block this neural damage by reducing the ER stress, and it is similar to the way the PDE4 inhibition works. You need to understand this thing in details. You can go through the reference that has been provided in the description. I hope you have understood big picture of this uh, images of these figures. There are two figures that are combined in this slide. So that's all for this video. Thank you for your time.